and Softbill Association. So very experienced aviculturists and very active in the world of aviculture and conservation. So if you just look. There you go. And if you'd just like to make Gary feel welcome, please.
Okay, so when Ivan first asked if I'd come down to talk, we got on to saying I'd be happy to give a couple of short talks, This is a short one, the second one is a longer one. But you'll see why it's very opportune to talk about the National Finch Census when I get right to the end. What I'm going to do is just quickly go through the outcomes of the last census that the NFSA undertook in 2020. Oops, I'll turn that on. Many of you be aware of what the NFSA is and what it's about. Your society is a member of the National Finch and Softbill Association. It's essentially a national voice for finches and softbills. And it's a body that aims to be proactive, not reactive to issues. And I really think for aviculture, that's what we have to be doing. We have to be ahead of the issues. And one of the key issues when NFSA was first formed back in 2006 was we didn't really have a good understanding of how many birds we have in aviculture. We know we've got a huge range of species available, so all the Australians and a whole range of exotic finches and allied birds based on the inventory that was available. But we didn't really know, what were the least abundant. That's the key question we had. Which species do we really need to focus on from a captive conservation point of view? There are lots of species like this that, well, I guess the silverbill is the favourite species for a few people here. Not many, yeah, for Richard. And I've still got silverbills, but many of these other species, you won't see any of these. I don't think there's too many of those around, but many species are declining and have been declining. And when we first started the census, we just focused on foreign species, and only 57 of the 78 exotic species that were supposed to be in the country showed up in that census, but the response was pretty poor. We don't know whether all of those species always were here or were never here really. But one thing that became very apparent is there are many species that were held by only a small number of people and were really vulnerable to disappear. So we had a whole raft of initiatives that were discussed back then about how we might conserve populations in aviaries, and you really need information in order to plan what you might do to target management. And so the first census was done in 2008. That was only exotics. But then we did one in 2010, and that brought Australian finches in, 2014, 2017, and 2020. And just to show you the sorts of the way the census runs, how many of you participated in a past census? Many of you. How many are going to participate in the next one? Everybody? The census is totally anonymous. Anyone can participate. You don't have to be a member of a club. You can put your entry in online. You can do it on paper. We don't want to know your name. All we ask is which state you're from. That's about the only piece of information. Initially, the response was pretty ordinary, but this is the sort of response we've had for the censuses up to 2017. You know, when I looked at that first, and at that time, Queensland Finch Society had close to 1,000 members, and nationally we only got 209 people to participate. It was pretty disappointing. But you can see the numbers have grown, and the number of birds that are captured in the census is becoming very significant. Now, 51,000 birds means that we're starting to get a reasonable picture of relative abundance of those species. We know we're not capturing everybody, obviously we're not, and we're not capturing all the birds, but we're getting a good picture. And the last census, that number went up by another 100. Another 10,000 birds were summarised in that census. So that's a reasonable picture, and you can see this is the number of respondents and the number of birds over the 5 censuses so far. But, 500, 600 respondents is only a fraction of the people that keep birds, and we really all need to be working on getting all those people out there who do keep birds, providing information into this census. It is totally anonymous, but the information is hugely valuable. In that census, this was the breakdown of responses by state. So you can see New South Wales, about 28% of the responses, Queensland, 26%, and so on. South Australia, Victoria, WA. There was a few people who didn't tell us their state, but I think it's great that whatever it was, 97% did, so that's good. And in those responses, about 80% of the respondents keep both native finches and softbills and exotics. We have some people who only keep natives, and most of those people are specialist zebra breeders or specialist Gouldian breeders, so that's all they have. And only a tiny fraction, 5%, only have exotics, and a lot of those are softbills. Now, you're not going to get much information from that, but you have seen a report on this last census I think you put it in your magazine, didn't you? But that just shows broad patterns for exotic finches over the 5 censuses. And it shows the common species that you would expect, the most common. And it shows, how the numbers have or haven't changed. In some cases, they're quite stable. In other cases, maybe they're more stable, they're fluctuating a bit. We see the common species are pretty consistent and largish numbers. So in that census, the orange breast was the most abundant exotic finch, for example, and I won't go through all of that list. But how we handle this data now that we've got 5 censuses and we'll have more, and with increasing number of respondents, we can't just look at total numbers for each species because that really doesn't tell us a lot. And just to illustrate why that is, if you consider, say, survey 1 is 2017, and for whatever this species was, we recorded 1,000 birds, and this is 2020, we got 1,500 birds. You might say, well, they've increased, 50% increase, that's good. But in survey 1, there were 200 respondents that had that 1,000 birds, and in survey 2, there were 500 who had those birds. That's actually a decrease in the number of birds per respondent. So it depends how you look at the data, and you really have to normalise the data for the number of people who respond so that you're comparing apples with apples over years. And so that's what this sort of data now is. And again, I know you can't read that, but this figure here is that normalised number of birds per species, so normalised by the number of people who responded in the census. And when you do that, you start to see some trends, and you might notice this species here, red-faced parrotfinch, over those censuses is actually declining in terms of numbers of birds relative to the number of people responding. I'm highlighting a few of those. 4 of those common species were declining over those censuses. If you look at the really rare species, well, you'd know what most of them would be. There are very few of most of these species left in the country. And again, I make the point, we know we're not capturing everybody in this census, so there are certainly people out there who have some of these rarer species and don't want to tell anybody. But, the rare species are pretty obvious, and these are species, particularly some of these that we really need to focus on if we're not going to lose them. Some of the species you'd be familiar with, red siskins, normalised numbers, relatively consistent. Yellow siskins, relatively consistent. Song sparrows seem to be declining. And we'll see what happens next time we do this. Munias, the four nuns, including the Javan munia, so black-headed, tricolours and white-headed. The white-headed munia in particular, a consistent decline in numbers, whereas tricolours perhaps are stable. And the Javan munia, of course, essentially collapsed. And whether many of these birds are pure Javan munias is always a question. So that's the sort of information we get from the census. There's some interesting trends for some of the waxbills. So orange-cheek waxbills starting to show a resurgence, more people with them, more people breeding, whereas the black-rump waxbill is essentially gone based on the census. And a few interesting things appearing, violet-eared waxbills appearing in the census. A few other species, again, the saffron, not many people keeping and breeding saffrons, oriental greenfinches. So these are all species that somebody needs to take notice of. Some of the species that were increasing for a few censuses, so the African silverbill, for example, increased when we had a fair bit of effort, but they're now dropping back again, and there aren't many left in New South Wales, unfortunately. Whereas Napoleon weavers seem to be on an upward trend, and hopefully will be a continuing upward trend. We get some interesting developments like species that weren't there in the first census and have appeared. They've come out of the closet or something's happened, but species, and some of these are starting to certainly not be common, like the number of people reporting these is only a handful, but they're present and they're in the census. If we look at native finches, a range of species, the most common native finch kept is the Gouldian. So these are Gouldians and Gouldian mutations, by far the commonest native finch kept in Australia. And the zebra is obviously second. And their numbers just highlight most of these common Australian species are pretty stable in aviculture. What else can we learn from a census like this? Well, one of the things you can learn, and this takes a little bit of explaining, but you can look at the frequency distribution of the size of collections. Because all the data is from individuals, we can look at how many people keep what sort of numbers of birds. And those of you at the front will notice a couple of these classes have got screwed up somehow or other in this figure. But essentially across the bottom here are people that keep only 1 to 50 birds, people that keep 50 to 100 and so on. So it goes up in 50 bird increments. And you can see the vast majority of people who responded to the census, about 40%, only keep 50 birds or less. And this line here is the percentage of all the respondents. So by the time you get to about here, up around 300 birds, that's pretty much captured all of the bird keepers in the country, but a big proportion of them have 150 birds or less. The smallest collections recorded were just 2 birds. 7 people reported 2 birds. And I think that's great that people that only have a couple of birds participated, because their information is perhaps not as important as some of these big ones, but it adds information, and every response is a piece of information. The largest collection in this census was 1,681 birds up here. The next largest was down here. a bit over 800 birds. So we get that sort of data. And the reason I'm talking about it is the next census is about to start. So the census opens on Friday. So you're the first club that has been spoken to about it. All of your club management team have, or at least Ivan has, have all the information. You have access to this online SurveyMonkey link. So the easiest way to put your data in is just to get that link, click on it, and you can just go through and enter what you have, or you can use a paper form. And so Ivan has a copy of the paper form that he can print for members. The census will stay open until the middle of December, so you don't have to do it in the next week. You can think about it. You might have to spend a week or so counting how many birds you've actually got. As I said, it's totally anonymous. All the data gets published. I think having this data is hugely valuable for our hobby. There aren't other parts of aviculture that have this sort of information, and it's easy to do. You can do it online, or you can put it on a piece of paper. So I would encourage you to participate in the census that will open. The SurveyMonkey site will be open. That's all I was going to say. You can have a cup of tea, unless there's some questions. Any questions? No questions? How many people are going to do the next census? All hands up. Very good. Okay, thank you for listening.
Thank you very much, Gary. Oh, no, this is very good. Time to hand over to Gary for him to slip into part 2 of his presentation this evening. Thank you very much, Gary.
Okay, thanks very much. There's some much better seats up here for all you guys over there. You won't be able to see anything. They always sit over there, do they? Okay. All right, I'll get underway. And before I start, just to thank you to the Society for the invitation to speak. And I meant to say earlier, a big thank you from Save the Gouldian Fund for the donation that you approved tonight. All the funds that we use to support research activities and conservation activities come from donations. And so that's greatly appreciated. Thanks very much. This talk is not an avicultural talk, so I'm not going to talk about breeding any of these species. It's a bit of a mixture talk. It's a bit of a travelogue. It's a bit of history, and it's a bit of science towards the end. And so it's really just to get you thinking and to expose you to some interesting facts from the past and from currently. So I've got 3 birds pictured up there, and the question is, how are they linked? They're 3 very different birds. Darwin's finches occur only on the Galapagos Islands, and they're thorpids in the Thorpidae. The Gouldian, of course, we know in Australia, and the Strildid, and the grass quits depicted here as by a Jacquerini, but it's only one of the Central American grass quits in yet another family, the Emberizidae. So they're not linked from the point of view of genetics or speciation. They're not closely related species. So how are they linked? And when I gave this talk once before, I said, so has anybody got any suggestions? And they essentially gave my talk for me. So I'm not going to ask you. But you may have some ideas. And I'm going to start with Darwin's finches. And that's because this talk really had its genesis in 2013 when Cheryl and I went for a visit to the Galapagos Islands. Fantastic. If you ever get a chance, any of you been to the Galapagos? If you get a chance, yes, if you get a chance, go. This is the Galapagos archipelago. And to go there is really a pilgrimage for someone like me, who's a biologist, ecologist, evolutionary biologist, because it's a key location that Charles Darwin visited on the Beagle back in 1835. Darwin only spent a short time, 5 weeks, in the Galapagos. He only visited 4 of the islands. There are many small islands. But that visit really helped him clarify the evidence that he slowly accumulated once he got back for natural selection as a driving force of speciation and evolutionary change. He didn't have a eureka moment when he was in the Galapagos. He spent that time collecting lots of specimens. The eureka moment really came later, but what he saw there really, as he reflected on it, sparked his thinking. And he speculated early on that what he saw there in terms of the tortoises that differed on different islands and the mockingbirds in particular might undermine the concepts at that time that species were stable, never changed, did not evolve. So he started thinking about that early. Some facts about the Galapagos. There are 18 main islands and over 100 small ones. It's part of Ecuador, and to get there, you have to go through Ecuador. It's on the equator, so it's an archipelago on the equator, 1,000 kilometers west of South America. So it's incredibly isolated out in the Pacific. And you can see the Galapagos there, 1,000 kilometers west of Ecuador. All the islands are volcanic in origin. And some of you would know how Hawaii was formed. Hawaii is on a plate that sits over a hot spot in the crust, and as that plate moves over that hot spot, islands are created. Well, exactly the same thing happened for the Galapagos. This is the Nazca tectonic plate, and it's slowly moving in this direction towards South America. And there's a hot spot under there that feeds magma up to the surface and progressively creates islands. And because it's moving in that direction, the islands on this side are the oldest and the islands on this side are the youngest as they're being progressively formed. And this big island, Isabella, and this one, Ferdinandina, are still very volcanically active. Whereas these islands over here are now no longer volcanically active and are being slowly eroded away, and they will eventually just disappear. So this is where the active volcanoes are. Sierra Negra is one that we climbed. Sorry, that's Sierra Negra. So 5 big volcanoes on Isabella. The oldest island that still exists above the ocean is Hispaniola, and it's 3.5 million years old. It's highly weathered, there's no water, and it will eventually just erode away. The youngest are these two, and they've got very active volcanic peaks. Across the whole place, there are no rivers and there's very limited surface water. So it's quite an extreme environment. Now, I've got an audio visual that Cheryl put together, but I think in the interest of time and because we don't have good sound, I'm not going to show it tonight. So we'll skip that. But just to show you, what some of the landscape looks like, this is in one of the craters on Isabella. That had been active only a month or two before we were there. But a lot of the landscape is very rugged, very dry. But progressively things start to establish, and there's, like this cactus that is growing directly out of the lava. The birds of the Galapagos, and obviously the finches are part of that fauna, is really very depauperate. There's very few species, a total of only 60 resident species. And you compare that to, say, Australia with, 860 something species. About half of that fauna is Darwin's, of the land birds, is Darwin's finches. So 60 resident species, many of them are marine species, aquatics, 26 land birds, and 13 of them are Darwin's finches. They're an incredibly interesting group of species from a point of view of evolutionary biology. They're probably the best example of adaptive radiation from a common ancestor that is known anywhere. The full group consists of the ground finches, Geospiza, these ones here, the large ground finch, the large cactus finch, the medium ground finch, the small ground finch, and so on. Then there's the tree finches over here, Camarhynchus, 6 of them, 6 Geospizas. There's the warbler finch, just 1. That makes up the fauna. The original species that colonized the Galapagos was the earliest land bird to arrive. And these species all radiated from that single ancestor. And that's what we're going to come back to towards the end. Who was that ancestor? Their taxonomy has always been a bit unclear. Originally, they were in the Emberizidae, that's the buntings and sparrows, but they're now classified in the thraupids, the tanagers, in a specific subfamily, but they're not related at all to true finches, although we call them finches. And they're very young species in evolutionary time. Those islands have been there for millions and millions of years, but each island is only there for a short time before it disappears. And these species, the common ancestor of these species, only colonized the Galapagos between 2 and 3 million years ago. that's very young. Now, one way you can look at the evolution of species and their relatedness is to look at tools like this. This is an online tree of life that essentially captures molecular sequence data. So you can get molecular data from all sorts of species and compare it and apply what's called a molecular clock and look at when different species diverge from one another. And if you move along the arms of this, you can progressively get down to species of interest to you. And so if we look at this arm, for example, right at the end is where all the passerine birds are. A huge part of the avifauna of the globe is just in this little arm. And as we go further down, what you see here are dates at which certain groups diverged from a common track. So all the parrots are captured just in this arm, and they diverged from a common ancestor 75 million years ago. If we keep moving down, we come to other species we might be familiar with, and keep coming, robins. We're going to keep moving right down here. Weavers, waxbills, and other things in here diverged 33 million years ago. The Australian grass finches, which I'm not talking about, are all in here. And if we keep going right to the end of that branch, we eventually get to this arm, which captures the tanagers, and Darwin's finches are in here. Finches and grass quits and other things. And this is the end of one of those little tendrils that shows some of those species. So the medium ground finch, about 4.5 million years, it's been distinct from this path. So if you're ever interested to have a look at this stuff, there's some fascinating information, and this tree of life is being added to constantly as more molecular information comes along. So I'm not going to focus too much more, but some of the other species like the large ground finch, large cactus finch, common cactus finch, really only diverged about 1 million years ago. So these are very young species. And they're interesting in the way they've evolved because you can easily notice the huge variation in beak size and beak morphology. And you can break them up very clearly in terms of, these are mostly the ground finches, in terms of what they feed on. So all these species feed on buds and fruits, seeds and parts of cactus or seeds. And these are mainly the cactus finches and the ground finches. And then the other main group feed mainly on insects. And so part of this, the evolution of these beak differences is due to diet, driving how these different species have evolved and specialized to survive in what is a really challenging environment for any birds. Some like these, you can see the huge variation in beaks. And shown again here, the relationship between size and morphology of the beak and the size of food sources for those species. So the large ground finch has the equipment to feed on large seeds. The small ground finch can only handle small seeds, and the medium ground finch sits in the middle. When food is really abundant, these differences in specialization don't matter much. There's plenty of food to go around. But the Galapagos, because it's such a harsh environment, goes through regular periods of extreme scarcity and challenge. And at those times, species that are specialized in some way to be able to preferentially use a subset of the diet, like the small ground finch or the large ground finch, will diverge. There'll be strong selection for that difference in beak size to be even further enhanced and stabilized. The end result, what we see in those species, those 13 species, is the end result of a complex process. There must have been some variation, slight variation to start with, and then selection, particularly in times of food shortage, and then inheritance of those differences. So there had to be a genetic basis for that. And so there was probably 4 key steps. Some geographic isolation among the common ancestor on different islands that led to some drift and eventual reproductive isolation, at least partially. When they came back together in what's called sympatry, there was then competition and pressure to further enhance those differences, those specializations through competition. So that's the sort of process that eventually led to that suite of species. And Darwin became really fascinated by this variation. He didn't become fascinated when he was there. He became fascinated later when he was back home. And that's an important part of this story. And it's, as I said, it's one of the clearest examples of adaptive radiation. When you visit the Galapagos, you will see the finches everywhere. They're incredibly common. It's sometimes very difficult to pick the differences between ground finches and tree finches, or the different sizes. You can certainly pick the difference between a small ground finch and a large ground finch, but the medium ones I found somewhat difficult. We saw lots of small tree finches feeding on flowers up in trees. The ground finches tend to feed mostly on the ground. We saw them down at the beach doing all sorts of things, feeding on vegetation. We saw them visiting marine iguanas and looking for parasites on their skin. We saw them taking rides on Galapagos turtles. This is on Santa Cruz Island. One of the islands that does have some surface water and gets a lot of rain, and that's where most of the population lives, the Ecuadorians that live in the Galapagos. They even come into some of the restaurants and check things out, so that's how common they are. In much of the dry landscape of the Galapagos, cactus is a dominant plant, and you can see them here growing on pretty bare lava fields. And so cactus is very abundant, and it's not surprising that 2 species at least have evolved to specialize in feeding on cactus. And this is the cactus finch, and you can see its extremely elongated bill. And here it's feeding on, and here they're feeding on open cactus flowers. But the importance of that adaptation, the elongation of the bill, is that it allows this finch to feed on flowers even when they're not open. It's able to pierce through the petals that haven't opened and get a feed at times when there's nothing else around, and none of the other finches can do that. And so that's the adaptation that allows it, particularly when food is scarce, to survive. You can see there it's got pollen all over its face. A lot of what it's getting out of those flowers is the pollen, and they eat the style in the flower as well. But there are some other amazing adaptations. This is one that we didn't actually see, but this is what's called the vampire finch. It's another one of Darwin's finches. They only occur on Wolfe and Darwin Islands, and there is no permanent water there. And they survive by drinking blood, which they get from wounds that they inflict on the gannets. And you can see them here feeding on blood oozing from wounds that they inflict on those birds. An amazing adaptation. There's also the woodpecker finch that uses tools, and you can see it here, using tools to extract insects. So it's one of the insect eaters. You'd probably be aware that tool use in birds is very rare. There are several parrots that use tools, but this is a great example of specialization to occupy a particular feeding niche that allows this species to survive, and it's had to evolve various morphological features that allow it to do this, as well as the behavior. You can see Darwin's finches down on the rocks by the sea, here feeding on what look like algae growing on those rocks. We could never really work out what they were feeding on. And as I said, the ecology and evolution of these finches has been the subject of a lot of research, and particularly by two people, Peter and Rosemary Grant and their students. They've written now a number of books, but all their work was based on this island just off the coast. In fact, it's just off where the airport is in the Galapagos on Santa Cruz. This is Daphne Major, and that island there is Daphne Minor. But all the work they did was on this island. And you can just see a bit of a track up the side here. The only access to that island is at certain tides, you can get on to a point here. And then they had to carry all their gear onto the island to conduct field research. They and their students worked on that island for 30 years, and they were there for 6 months of every year. And you can imagine how challenging that was. And they focused their work on the medium ground finch, just one species. They followed every individual bird throughout their lives, and they don't live 30 years, so they were following several generations during times of highly changeable food availability. And what they were able to see was rapid evolutionary change happening in that 30-year period in response to strong selection imposed by changing food availability. It's a fascinating study, and it's really captured very well in this book, The Beak of the Finch. If you ever want to read a great book, that's one. And Peter and Rosemary Grant, that's them that's where they lived for 6 months each year. Not just them, they had students on the island, and they put together this book. I think that was released about 5 years ago. Fascinating stuff. Now, as I said, Darwin was fascinated by these finches, and he made some really prescient comments. And I'm not going to read all of this, but essentially what he's saying here is that he's seeing this change in structure of just one group of birds in response to selection happening. And he was starting to think that this can only have occurred through one species diverging into all these different ends. A very different concept to what applied at the time, which was that species were fixed and never changed. And some of you, I remember I organised a Darwin birthday party in 2009. It would have been his 200th birthday. On that day, an analysis of the full genome sequence of one of Darwin's finches was published, and the great thing was that it showed that among the 13,000 odd genes that code for proteins in that genome, there were many that were subject to positive selection, but two in particular involved with beak morphology, that if Darwin had been around now, he would have been over the moon to see that sort of genetic evidence of selection impacting on beak morphology. So we come to the first bit of the question then. So what's the link of these species that I've just talked a lot about and this one that has nothing to do with them? Now, does anyone have a guess? No? The link is John Gould himself. So it's a link of an individual between these two species. Now, what is the basis of that? Well, you all know John Gould. You know him as the father of some amazing books of lithographs of birds, not just Australian birds, but lots of birds all around the world. 21 books Darwin and his group produced, 3,000 magnificent lithographic plates. He was a successful businessman, and he set about producing these monographs to make money. So he saw that there was business in nature. And he was called the British Audubon, after, of course, Audubon in the US. But the interesting fact is that that Gould didn't paint any of those pictures himself. He was not an artist, even though his name is always associated with all these things. He didn't produce them. The artists were largely his wife and a suite of other very talented people. What Gould did do was do early sketches of each of the birds, and he did sketches of the habitat that you might see them in, some of the plants, and provided that to the artists, but they then did this work. And Gould played a really pivotal role in how Darwin interpreted what he saw in the Galapagos. When the Beagle returned to England in early 1837, John Gould was the foremost ornithologist of the time, and Darwin gave him his whole bird collection to look at. Darwin wasn't really all that interested at the time. He thought it was just a collection of all sorts of different things. But Gould very quickly, so this was the 4th of January, by the 10th of January, Gould came back to him and said, "This is brilliant. What we have here is a series of finches that are so peculiar, form an entirely new group of 13 species." And this hit the headlines of the newspapers. So Gould was just, couldn't believe it. And he enthused Darwin to then start thinking a lot more about these species. So by March, Darwin got a full report from Gould of all the birds. He classified all of them. Most of them were new and distinct forms, obviously allied to South American bird fauna. But Darwin's interactions with Gould were pivotal in how he then started to think about this suite of species and formulate his ideas about natural selection. So Gould was really very important in that important breakthrough that Darwin eventually had. And of course, we know Gould as the person who named the Gouldian finch and named the bird after his wife. And so it's often called the Lady Gould. He named it after his wife because she was so critical to his success, particularly when they were in Australia. Elizabeth did enormous amounts to support him in Australia. As it says here, she laboriously assisted me, and so he named Gouldian after her. So that's the link of Darwin's finches and the Gouldian, is through John Gould. So where do the grass quits fit into this story? John Gould didn't have anything to do with them. Where they fit in is when we ask the question, what was the common ancestor of Darwin's finches? And you can start to answer those questions by, again, looking at molecular evidence, looking at genetic sequence data, and trying to understand what is the closest living relatives to this species. So there are a whole range of related species, all tanagers in South America, like this stunning bird. This isn't related, of course, but another stunning South American bird. But how do you measure the level of relationship between these species? Well, the best way, or any of these species, is through looking at DNA. Some years back now, this really started in the mid-70s, but has progressed enormously. But techniques to start extracting DNA and hybridizing DNA from different species. So the way people started looking at how similar is the DNA of species X and species Y. They'd use a technique called nucleic acid hybridization, and essentially they're looking at how you can get different strands of DNA from one species to match up to another. So you take DNA from the two species, you heat it, it breaks those bonds, and you end up with single strands, and then you look at how readily can the one strand from one species pair with the strand of the other. And the number of successful connections shows you how related those two species are. If you get essentially no connections, then they're highly unrelated. But in very closely related species, there'll only be a few places where a strand from this species and from that species won't pair up. Now that's how it used to be done. These days it's done in a black box. You just extract DNA, you put it into a machine that does a full sequence, usually just of genes from mitochondria, not whole genomes. And you can just compare the sequence similarity and say how closely or otherwise one species is related to another. And that's what lots of people have been doing, and they produce these phylogenetic trees that show the similarities or divergences of different groups. And this is quite a wide distribution of species, but some of them you would know, of families, sorry. So the Placeids, like the African weavers, and the Astryldids, like our grass finches, are very closely related. So when you look at these diagrams, if you've got two groups on adjacent branches, they are very closely related. If they're way on another branch, they're much more distantly related. And the distance from this side tells you something about the time of divergence. So our grass finches and African weavers are very closely related genetically. The sparrows are somewhat more divergent. And then the true finches, the fringillids, are not related at all, really. They're highly divergent. And so that's the sort of technique that's used to ask the question, how related is a species like the Cuban, which is a grass quit, to Darwin's finches? So lots of studies have now been done doing exactly that. And the early studies suggested, in fact, that the Jacquerini was the most closely related existing species to Darwin's finches. But then more work was done. And this is probably the most seminal paper comparing different grass quits to Darwin's finches. So the Cuban you'd know, and the Jacquerini, this is the bicolored grass quit, and this is the dull or obscure grass quit. And this is it's not a cladogram, it's a phylogenetic tree from that study that Sato and his partners published. This is all Darwin's finches here. They all aggregate very closely together. And this is the most closely related existing species. The obscure or dull-coloured grass quit is the nearest living relative to Darwin's finches. They diverge very, very little. When you start doing molecular comparisons, you use about 4% divergence to say they're different species. If there's less than 4% divergence, you say it's the same species. So they're only 4.6% different. And so it suggests that not that this was the original ancestor, but that this species and this suite of species had a common ancestor. And using a molecular clock about 2.5 million years ago. So that's where the grass quits fit in. The Gouldian finch links to Darwin's finches by that personal link of John Gould and Charles Darwin. And the grass quits fit in because there's an evolutionary link there in that the grass quits share a common ancestor with Darwin's finches. There is no link that I can find between grass quits and Gouldians. So there isn't we can't close the loop on this story, but we're at the end of the story. I just want to acknowledge the inspiration that Darwin brings to most biologists. Cheryl for her fantastic photos that I used throughout, but were really in that audio visual that I didn't show. And Jonathan Weiner wrote The Beak of the Finch. So if you want a good read, it's worth looking at that. So thanks very much. Thanks for listening. I hope you found it interesting. It's not going to help you breed lots of birds, but I've just tried to bring out some interesting relationships and explain a bit about how these fantastic species evolved. Any questions? Would have been a very small population that got there, yep. Yeah, probably a storm of some sort. Yep. They would have been a very closely related small population to start with.
Sorry, we tend to look at things on what they physically look like when we say that they're diverged, but also their characteristics on their behavior changed as well. I look at each of those. Now, when we look at those cactus ones and that, the large beak, the small beaks, do they interbreed between those groupings? Did they notice that at all? That they're, because genetically, if they're coming down from the same line, they possibly could. Like we even see that with our birds here.
There's no evidence at the moment of any interbreeding of the cactus finches with the ground finches.
Yeah, that's
But within the ground finches, on Daphne minor, they certainly did find evidence of the medium ground finch and the small ground finch hybridizing. That didn't happen very often, but it generated some new variation. And
yeah.
you know, that's really important for ongoing evolution.
But is that perhaps how the first breakup came about? Just a few birds started to find not a physical difference, but a different food source when they were starving, and therefore they evolved into different.
Inevitably, though, it would have started with isolation on different islands. So small population, a few birds got to an island and then were isolated for a long time, and slow divergence while they're separated. And they'd be specializing in different ways on those islands.
Yeah.
Anyway, it's
Thank you.
and great to read that book of the Grants, again, if you're interested.
The earlier presentation, you were talking about the census, and that we lose birds, and we often lose them because they're inbred. And these things have been doing it for 2.5 million years with a handful of original parents, and
they started with a small initial pool, but then they would have spread slowly to different islands and built up populations that then diverged. When they came back together, that's when you would have got a spark of variation happening. You got any hybridization. So yeah, you know, trying to all sorts of ways to build genetic diversity in these populations would have been critical. They would not have survived if they didn't.
Yeah, that's what
If they had been just a handful of birds and they didn't change, they wouldn't have survived.
I've often heard people say that the island species, not just Galapagos, but any island, are less prone to inbreeding than other things, much like reptiles. You know, they come from a very limited place, but they can interbreed.
But on islands, most of those, you find most of those populations are very closely linked.
Yeah, but they don't disappear, you know,
They don't disappear, no.
Yeah. Anyway, thank you.
Okay, no other questions? Thanks for listening. I hope you enjoyed it.
Okay, thank you very, very much, Gary. That was a very informative talk. We've got a little thank you for you that we haven't got here this evening, so we'll be posting something out to you. All right, so it's time for the raffle. Over to you guys up the back there.